Hello, everyone, and thank you once again for joining us on this first installment of a series of webinars hosted by Aging Analytics Agency, focusing on a discussion of the current state of healthy human longevity and the translation of developments in geroscience and the science of longevity into clinical practice. Today, we will be hearing from Dr. Arkady Prokopov, an integrative medicine physician and CEO of Athletic High Tech SL, with over 30 years experience in biogerontology generally, as well as the effects of intermittent hypoxia and hyperoxia on mitochondrial aging and human longevity in particular. Today, Dr. Prokopov will be delivering a keynote lecture on the history and current state of a broad line of research focusing on the impact of intermittent hypoxia and hyperoxia on human aging and longevity, and his specific interpretation of the validity of a recent clinical research study's results, which claim to have facilitated some degree of human rejuvenation as a result of intermittent hyperbaric oxygen therapy. Thank you so much for joining us for today's event. Dr. Prokopov, the floor is yours.
Thank you very much, Franco. Yes, I'm ready for presentation. I just want to say a couple of words that this article of Israeli scientists, of course, it's made a great resonance and the media hype, and contrasting opinions were presented. We will discuss result and key points of recent hyperbaric oxygen therapy paper. We will discuss physiology of hypoxia-hyperoxia and mitochondria as drivers of senolysis. Also, we will speak about COVID-19, mitochondria, and endothelium in this disease. Then we will discuss, we will see overview of oxygen-related mitochondrial interventions. And we will focus on controlled intermittent oxygenation. And then we will speak with Dr. Dmitry Kaminsky about comparative and SWOT analysis. Clinical experience often goes ahead of scientific research. Detailed explanation comes sometimes much later, when a successful method is already popular. But now I have a challenging task to deliver my view of the problem to both scientific community and laypersons. Today we are discussing biomedical technologies that are routinely used in clinic already for decades. These technologies employ a controlled change of the amount of oxygen in a cell. By increasing or decreasing oxygen partial pressure in the tissues, we can influence the fate of cellular organelles and cells. Ultimately, we can either accelerate or slow down the aging process in the body. This is the paper of Israeli scientists. They recently published a paper which attracted many critics from scientists and lots of media hype. Researchers proclaimed that hyperbaric oxygen treatment resulted in telomere lengthening and reduction of senescent cells in senior participants. The HBO protocol they used comprised of 60 daily sessions, 5 sessions per week, within a 3-month period. Intriguingly, many beneficial effects that have been achieved usually occur in physiological hypoxia that happens during embryonic development, as well as in certain lifestyle interventions. The researchers claim significant senolytic effects and telomere lengthening. Lots of criticism from gerontology community is related to statistic analysis and interpretation of underlying molecular biological mechanisms. So let us take a deeper look at this problem. Being a practicing physician specialized in integrative mitochondrial medicine, And being a gerontologist, I also have the background in aviation, space, and diving medicine. I worked a lot with both HBO and hypoxic interventions. Relying on my experience and referring to the published scientific research, I personally see no contradictions and no serious bias in this study. The key word here is a repeated intermittent HBO exposure. Large evidence is accumulated that both hyperoxic, such as diving, and hypoxic, altitude or simulated altitude interventions produce telomere lengthening. But why then also lifestyle interventions are beneficial? Do telomeres really grow like plants by watering? How hyperoxia may overlap with hypoxia? What exactly is the difference between both, and why the oxidative road looks so attractive? In very simple presentation, in very simple image, we can see that action of a toxic damaging agent can cause stress, damage, insufficient adaptation, and cytotoxicity. But if the same agent is given in small doses repeatedly, it causes adaptation response. It causes protection and induces homeostatic regulation. It is a universal phenomenon called hormesis, and of course we should take into account this possibility in this study. Let us take a deeper look at hypoxia and hyperoxia physiology and underlying molecular biology. A closer look at HBO mechanisms shows that in this protocol, the HBO employed breathing 100% of oxygen in a hyperbaric chamber. Venting out exhaled gas mixture prevented increase in the oxygen in the barochamber. And intermittent exposure to varying oxygen concentration caused pulsating of oxygen partial pressure in the tissues, cells, and mitochondria. This initiates oxidative chain reactions at cellular and mitochondrial membranes, which can deplete mitochondrial antioxidative defense. But here, oxidative stress has been prevented by normoxic exposures, facilitating recovery of endogenous antioxidative system. During hyperbaric oxygenation, the reactions which take place, of course, they're universal. Hyperbaric oxygen causes oxidative stress if it's a constant, uninterrupted hyperoxic exposure. Usually, this manifests as lung or CNS toxicity. However, I remember that while conducting my research in divers in 1980s, I have been achieving up to 45-60 minutes continuous exposure at 4.0 ATA, breathing 100% oxygen without significant toxicity. I've done this by combining antioxidative supplementation and the intermittent hypoxic preconditioning. On the other hand, a continuous or intermittent uncontrolled hypoxia, such as found in sleep apnea, also depletes enzymatic antioxidative system, causing multiple pathophysiological effects routinely found in various acute and chronic diseases and conditions. In their earlier paper, the same Israeli authors argue that repeated intermittent hyperoxia induce adaptations that produce the same benefits as those seen in repeated intermittent hypoxia. They call it the hypoxic-hyperoxic paradox, which creates cross-adaptation to both hypoxia and hyperoxia and also augments cellular resistance to a number of many damaging factors. How is this possible? The HBOT is successfully used in clinic for decades. Evidence accumulated that the majority of HBO molecular mechanisms rely on mitochondrial hormesis, which can explain beneficial changes and curative effects of HBO treatment. Here is an example with traumatic brain injury. The authors say that HBO-induced neuroprotection, in addition, reduction of lipid peroxidation and upregulation of antioxidant enzymes are among the mechanisms involved in the action of HBO. In that way, HBO diminishes imbalance between oxidants and that occur after brain injury and contributes to the maintenance of pro-oxidant homeostasis. Pro-antioxidant homeostasis. An emerging body of evidence indicates that the generation of free radicals by mitochondria plays a critical role in damaging cellular components and initiating cell injury and death. On the other hand, HBO-induced mitochondrial reactive oxygen species have been shown to participate in preconditioning by influencing calcium-activated potassium channels. Pulsating mitochondrial ROS activate cell survival programs that render tissues and organs more resistant to the damaging effects of ischemic reperfusion. This may explain various therapeutic interventions which selectively employ mitochondrial ROS production to reduce and repair tissue injury. Here we can see that throughout the oxygen cascade from lungs to mitochondria, the oxygen partial pressure drops dramatically. So from 160 millimeter quicksilver down to 3 till 10 millimeter quicksilver. At normal conditions, PO2 is stabilized at mitochondrial level on this very low partial pressure. This corresponds to the parameters existed about 2.5 billion years ago, when symbiosis between anaerobic cells and oxygen metabolizing alpha proteobacteria occurred. So the oxygen cascade components and mitochondria conspire in order to maintain an unsigned oxygen level in order to optimize OXPHOS and ATP production, and also to reduce oxidative damage to the mitochondrial genome. But an extreme hyperoxia does induce metabolic disturbances and damages mitochondrial DNA via excessive ROS production. This excellent map of metabolic and signaling pathways underlying human aging is created and constantly updated by John Farber. Many thanks to him. Can it help us to elucidate why and how does cross-adaptation to hypoxia and hyperoxia occur? Let us look at it through the magnifying glass of the updated Darwinian model of the biological evolution. Today, we believe that life, as a matter of fact, is a competition and cooperation of DNA molecules in order to survive together and relocate their teams into the next generations. This requires a continuous, uninterrupted adaptation and dynamic adjustment to changing environment. The coordinated activity of all these complex pathways and molecular mechanisms is fueled by the ATP energy, 95% of which is created by mitochondria. We have here two collaborating genomes, two teams of genes that strive for mutual success. Nuclear genome cares about structural resources and stabilization of cellular environment, and mitochondrial genome provides energy flow on actual demand. And their mutual goal is not a prolonged and healthy lifespan. All these can be easily sacrificed by both genomes in rivalry with their peers to achieve the final destination, a successful transfer to the next generation. Do the shock waves that run through this network and caused by oxygen and ROS oscillations can influence biology of aging, the tempo of aging? We can understand a lot from this map, which is created by Douglas Wallace. And it presents the role of mitochondria, which is crucial in both embryonic and post-embryonic development for the maintenance of adult tissue homeostasis. Primary mitochondrial defects or alterations and pathways underpin a large number of human diseases, including neurodegenerative diseases, metabolic disorders, cancer, and also premature aging. Functional variants of mitochondrial DNA, polymorphisms, provide efficient adaptation to contrasting environments, such as hot desert and ice-covered polar seas. But if we mix them together in one cell, the resulting dysfunctions can lead to disease and accelerated aging. Let's take a look how mitochondrial change ROS production when oxygen content is fluctuating. A common feature of all animals is exercise. In other words, functional muscular activity, which is a major driver of reactive oxygen species. Exercising creates intermittent ischemia, in other words, intermittent hypoxia, and is known to induce mitochondrial biogenesis, mitophagy, mitochondrial regeneration. Hypoxia-sensitive calcium and potassium channels in the inner mitochondrial membranes play a leading role in this process, regulating the uncoupling of mitochondrial membrane potential, controlling proton leak, and preventing excessive ROS production. The contrasting effects of constant and intermittent hypoxia are clearly seen here. If we take a look at the molecular level in the mitochondria, we understand that pulsating ROS act as a double-edged sword, stimulating antioxidant enzymes in hypoxia. It is shown that under mild hypoxia, controlled intermittent hypoxia, which happens during muscular contractions, synthesis of superoxide dismutase, peroxidase, catalase, and glutathione peroxidase, glutathione oxidase increases. At the same time, an ADH is also increased in hypoxia. But in hyperoxia, we see the consumption of antioxidative enzymes. Pulsating ROS may drive mitochondria quality control. Spontaneous episodes of functional reduction of blood flow are found in all healthy tissues, not only in contracting muscles. They induce pulsating hypoxia, which correspondingly produce ROS oscillations in mitochondria that influence mitochondrial fission, fusion, and mitophagy. ROS activate the cellular surveillance system that protects its mitochondria from oxidative stress. These are antioxidative enzymes which prevent buildup of ROS beyond permissible levels, chaperones to refold misfolded proteins, and proteases to degrade damaged proteins. In case the mitochondria is beyond repair, the entire organelle opens and releases caspases that may cause apoptosis. This intrinsic mitochondrial quality control machinery is ROS dependent, ROS modulated. It constantly eliminates most damaged mitochondria, purifying mitochondrial populations in the functionally active cells and tissues. Now back to telomeres. It is clear that observed in the HBO phenomenon of telomere elongation is obviously caused by the ROS-modulated apoptic elimination of circulating senescent immune cells, having shortened telomeres. At the same time, maturing immune progenitors with longer telomeres gradually replace them. We see here that the longest telomeres are found in pluripotent stem cells in very early stages of embryo development. And then adult stem cells, they already have stable telomeres, but all differentiated somatic cells, they undergo telomere attrition. Here we see a mother of the all circulating red blood cells and white blood cells, the pluripotent progenitor. And as soon as they differentiate, they started to perform their functions, they undergo oxidative stress, and they shorten their telomeres. Of course, these cells, when they undergo aging and oxidative damage, they are more sensitive to oxidation, and they can be destroyed by strong oscillations of oxygen partial pressure. Senescent circulating immune cells are on the front line. The residential senescent cells in critical tissues such as muscles, heart, and brain are better protected from oxygen pulsations and can respond later. But we can see that ROS-inducing interventions potentially would induce or facilitate systemic senolysis. So HBO does not elongate telomeres, but facilitates elimination of senescent cells with shorter telomeres. Mitochondrial ROS can drive aging process. An uninterrupted and prolonged hypoxia or hyperoxia exhausts antioxidative system, hinders antioxidative enzymes build-up, and prevents supercompensation of antioxidative system. And if the system is overwhelmed, the mitochondria unfolded protein response is activated to increase the synthesis of structural mitochondrial proteins. This is mediated by the bidirectional crosstalk among the entire mitochondrial network, as well as between mitochondrial DNA and nuclear mitochondrial genes, which are relocated in the cell nucleus for millions and millions of years ago. So mitochondria actually work in cooperation. Here we find genes that are responsible for actual energy demand, and here are the structural genes which respond much slowly to supply damaged proteins in mitochondrial structure. So both hypoxia and hyperoxia, when prolonged and uncontrolled, can accelerate mitochondrial aging and shorten healthy lifespan. Controlled intermittent oxygenation can improve mitochondrial quality. There were many publications, research papers on this topic. This is one of them from 2000, that intermittent hypoxia exposure prevents mitochondrial deletion and mitochondrial structure damage produced by ischemia reperfusion injury. Exactly, this phenomenon can be explained by hormetic response. Hormetic response by intermittent hypoxia and hyperoxia stimulate the same ROS-driven mitochondrial response, which can be either protective or destructive depending on protocol. Mild stress stimulate hormesis through adaptive ROS signaling, autophagy, unfolded protein response, and heat shock proteins. It causes preconditioning state, which increases stress resistance and cell survival. If no preliminary adaptation, no hormetic intervention, then cellular damage occurs after a strong application of the same mechanism, in our case, high pressure of oxygen. Of course, it is also shown that chronic intermittent hypoxia can trigger a senescent-like phenotype, not only in human white preadipocytes, it can be also seen in neurons. But on the other hand, hypoxia prevents mitochondrial dysfunction and senescence in cardiac progenitor cells. We can see that hypoxia is also a double-edged sword. Oxygen can be a poison and can be a medicine. The end results depend on the partial pressure of oxygen and timing of oxygen oscillations. Again, a controlled intermittent oxygenation is the key here. Now let's talk about COVID-19, which is extremely important now, and mitochondrial dysfunction and mitochondrial aging do influence COVID-19 morbidity and mortality. It is shown in many research papers that mitochondrial polymorphisms, mitochondriopathy, or a chronic disease or age-related mitochondrial dysfunction can explain the vast differences in morbidity and mortality in COVID patients. We see here that healthy mitochondria correspond to low inflammation, balanced immune response, and high specific values of immune cells. Aging induces damaged mitochondrial damage, which is known to induce inflammaging, immunosenescence, chronic inflammations, and of course, deficient antiviral response. Improvement can come from inducers of mitoautophagy, caloric restriction, bioactive compounds, physical exercise, and of course, mitochondrial supplementation. The specificity of COVID is that it damages the endothelium in the vessels. Endothelium is which forms the inner cell lining of all blood vessels and lymphatics in the body, is a spatially distributed organ of critical importance for metabolism. The endothelium weighs approximately 1,000 grams in an average human and covers a total surface area from 4 to 7,000 square meters. And endothelial cells are extremely vulnerable to coronavirus infection. What happens then when endothelial damage occurs in various organs? Endothelial damage underlies disseminated microvascular coagulopathy. Regional variations in blood perfusion and differences in local endothelial damage can explain the multitude of pathology and symptoms. Mitochondria decline. Mitochondria energy production declines in aging. We can see the chart which shows the mitochondrial maximal oxygen consumption VO2 max. It's in untrained person. This is in different athletes, but nevertheless, athletes physical exercising can help to prevent abrupt mitochondrial decline. Nevertheless, it happens. In any way, it happens, and we see that it influences the most important clinical symptoms, such as loss of bone mass, loss of gray matter, and immunoaging. So osteopenia, sarcopenia, immunopenia, and neuropenia as age-related depletion of stem cells, accompanied by apoptosis of senescent cells. There are many different manifestations of a universal systemic process, which is mitochondria driven. Here we see the decrease of volume of brain, gray matter, so neurons in the brain. And here's the explanation why and how senescent cells are undergo apoptosis. Apoptosis acts as a natural senolysis mechanism. It is a cancer protective mechanism in the most tissues of humans. As we see, young tissues have a substantial amount of stem cells, and they residential stem cells and circulating stem cells, such as stromal bone marrow cells, and mitochondrial ROS damaged cells will be gradually eliminated. But age-related depletion of stem cells results in the appearance of functional threshold, and all related tissues undergo degradation process, degeneration. Therefore, it is obvious that mitochondria, immune aging, and endothelial aging are mitochondria dependent. Hypoxia can work in the process of in the COVID pathology in both ways. So we know that many damaging effects of COVID infection produced by hypoxia, by regional hypoxic ischemic damage to the tissues. But on the other hand, hypoxia, if used properly, can help first to prevent COVID, and second, most important, to accelerate regeneration after COVID infection. We now have success with so-called COVID long haulers using HBO, and we have very good success with intermittent hypoxic applications. So if we can summarize oxygen-related mitochondria interventions, HBO cause powerful pulsating of oxygen partial pressure in the tissue, cells, and in mitochondria. This initiates oxidative chain reactions in cellular and mitochondrial membranes, stimulating mitophagy and mitoptosis that precede to apoptosis. Senescent cells have most ROS-producing aged mitochondria, so they undergo apoptosis in the first line. So HBO-created oxygen oscillations facilitate apoptosis of senescent cells. And this explains senolysis of immune cells, telomere elongation, which comes from younger immunocytes that come from bone marrow, and systemic senolysis, which was discovered by Israeli scientists. But what happens on the other side of the coin? We find oxygen pulsations at the very beginning of mammalian embryo development. They govern the embryogenesis and control the intrinsic developmental senolysis via ROS modulation. Mitochondrial improvements caused by physical exercising employ the same pattern using PO2 oscillations at the mitochondrial level. So we see that on the mitochondrial molecular level, oxygen oscillations mitochondria sense oxygen oscillations irrespectively, is it from natural oscillations which happen during embryonic development or physical exercising, or probably from other interventions. What will happen if we reintroduce oxygen oscillations in adults? Can we reactivate the embryonic developmental and regenerative mechanisms in adults using controlled intermittent oxygenation? This problem was empirically solved and used already many years ago. Here we can see the therapeutic barrow chamber, hyperbaric chamber for therapeutical use. And it is used in clinics. In short, physiologically limited and controlled hypoxia contributes to numerous beneficial cellular reactions. And there are many technical solutions for this, different barrow chambers. By the way, this barrow chamber also is used for hyperbaric oxygenation, so it can work in both ways, for hypoxic treatment and moderate hyperbaric oxygenation. The first substantial work, substantial clinical research on application on hypobaric hypoxia was Mr. in Russia. Later, he published his results in the United States. We can see here that all important physiological and therapeutical indexes, the indexes which actually determine the health level, physical performance, they improve after adaptation to hypoxia. He described also protective effects specifically to cardiovascular diseases. We see here that intermittent hypoxia induces increased power of the oxygen uptake and transport systems, which leads to enhanced tolerance to hypoxia, ischemia, and physical loads. Activated brain RNA and protein synthesis, formation and fixation of temporary connections, which leads to enhanced resistance to stress damage. Audiogenic epilepsy, it's a model used with rats, and so on and so on. So it's a very substantial work. If you have interest, you can find his monograph in the United States, and it's very detailed and deep analysis. But now we have, in detail, this analysis shows that the same pathways are activated in normobaric intermittent hypoxic intervention. It is hypoxia-inducible factor modulated expression of genes which mediate preconditioning benefits via nitrous oxide and nitrous oxide synthase pathway. We find here increased mitochondrial biogenesis after intermittent hypoxic-hyperoxic treatment. We see increase of mitochondrial metabolism. We see improvement of pH regulation. We see significant reduction of oxidative stress. We see the increased expression of mitochondrial and antioxidative enzymes, except glutathione peroxidase. Glutathione peroxidase is lower after hypoxic adaptation. It looks paradoxically, but we should understand that glutathione is the most required antioxidant, cellular antioxidant, and if oxidative stress is lower, then less glutathione will be consumed. Therefore, the activity of glutathione peroxidase is lower. All these phenomena happen in athletes when they undergo altitude training. And now many athletes use instead adaptation to normobaric intermittent hypoxia. So the controlled physiological intermittent hypoxia reoxygenation episodes are the key mechanism of hypoxia and hyperoxia adaptation. Clinically, it is used already. It's sometimes it's just investigative clinical research, but nevertheless, in some clinics, it is quite well-known protocol, normobaric hypoxic treatment, and it's used virtually in hundreds of clinics and sanatoria all over the world. Many monographs are published on this topic. Here's one of them from 2012, and let's take a look on the content. Experimental use of intermittent hypoxia in Parkinson's disease. Protective effects of adaptation to hypoxia in experimental Alzheimer's disease. neuroprotective mechanisms of intermittent hypoxia in vitro study. So these mechanisms are really deep investigated in molecular and cellular models. Hyperbaric intermittent hypoxia is still used as method for improving functional state of pilots and astronauts. Intermittent hypoxia improves male subfertility. We have at least one evidence of anti-cancer effect of intermittent hypoxia in acute myeloid leukemia. Energotropic effects of intermittent hypoxia, the role of succinate-dependent signaling, very deep research from Professor Lukyanova in Moscow. And here is my article about intermittent hypoxia and health. It's just a review. Later I will show some illustrations to this. There are many clinical manuals and guidelines which are used since 1980s. Mitochondria are recognized as a target for intermittent hypoxia. And it is started to be used in mitochondrial medicine. We apply intermittent hypoxia, for instance, for treatment of autism. And children in the age from 2 years old, they already are able to get benefits from this treatment. On the other hand, senior persons up to 90+ years old, they also benefit from the same treatment. Here's some illustrations from my article, which help to understand what's happening during this procedure. First of all, mitochondria mutate very quickly, as you know. That's tempo of mutation is on magnitude higher than nuclear gene mutations. And mitochondrial DNA, which is a ring, it after the insufficient repair, it becomes smaller and smaller. And smaller molecules make their copy much faster than the larger molecules. Therefore, it is a phenomenon of clonal expansion of mutated mitochondrial DNA. We see these mitochondria as megamitochondria. You can find them in the cells, especially long-living cells like neurons and cardiomyocytes. And that happens under stable oxygen supply and ad libitum nutrition. But reactive oxygen species, here is just mutation, how mutations on the normal, so to say, mitochondrial metabolism, it's inevitably causes clonal expansion of mutated mitochondria in the cells. Clonal expansion leads to production of senescent cells. And introducing oxygen oscillations, controlled oxygen oscillations, that's very important. We can influence this process. So the healthy, undamaged mitochondria survive these oscillations, but damaged megamitochondria will be destroyed. Here I just called this intermittent oxygen restriction in parallel to intermittent calorie restriction, because both interventions have the similar molecular biological mechanism, so they are very deeply related on the mitochondrial level. After many series of treatment with hypoxic intervention, with controlled hypoxic elimination of damaged mitochondria, we see repopulation of long-living cells like neurons and cardiomyocytes with healthy mitochondria. Senescent old mitochondria will be eliminated, and younger undamaged mitochondria quickly repopulate cells. To summarize, this is the picture which shows the connection between extreme hypoxia and physiological hypoxia. Intermittent hypoxia, yes, it can be extreme, like in sleep apnea. And if the stress load, oxidative stress on the cells, on the mitochondria is too high, there will be not enough time and resources for regeneration. And so we see the increase of damaged mitochondrial DNA, increase of megamitochondria and senescent cells. Physiological hypoxia eliminates senescent mitochondria, damaged mitochondrial DNA, and we see improved mitochondrial quality in the cells. The same phenomenon, mitochondrial hormesis, can explain these reactions. These reactions are shown in the different models, cellular models, and in vitro, and also in animals. In humans, of course, we cannot so easily take mitochondria for analysis, but clinical effects can be explained by this. Let's now go to key points of intermittent oxygenation. So it is composed of two different possibilities. Controlled intermittent oxygenation is a generalized denomination of both hyperoxic and hypoxic interventions that induce or can induce mitochondrial hormesis. And the underlying molecular mechanism is the same. It's elimination of damaged mitochondria. And of course, on the first line, the most damaged mitochondria, most mutated mitochondrial DNA will be eliminated. The patterns of controlled intermittent oxygenation in general, we see that they originate from the normal embryonic hypoxic cycle, which takes place during the embryonic development. And it continues till the delivery. So this process is very, so to say, is very fundamental. It is found in all mammalians. Therefore, if we reintroduce this process in artificial way by inhaling hypoxic breathing mixture, we reintroduce this process in adult cells, in post-embryonic development, and adult ontogenesis. It is hypothesized that natural intermittent oxygenation governs embryogenesis and provides mitochondrial quality control. Controlled intermittent oxygenation as a naturally based but engineered mitochondrial rejuvenative interventions augments and supports developmental and regenerative mechanisms. The pattern applied can look very different. The first attempts were purely with hypoxia and normoxia. There were also quite successful applications of only hyperoxia, especially during physical load. That was done by Manfred von Ardenne in Germany. And now we combine both. We combine hypoxic intervention and interspersed with hyperoxic interventions. And both, of course, are designed in a physiological window. So we don't damage, we don't cause excessive damage. Practical applications are numerous. Here you can see the paper from German scientists who engaged in this field. This is application of intermittent hypoxic-hyperoxic training in geriatric patients. 34 patients from 64 to 92 years old participated in this randomized controlled trial. It was actually a multi-component intervention. They also underwent physical training, and the results: intermittent hypoxic-hyperoxic training turned out to be easy applicable to and well tolerated by geriatric patients up to 92 years old. This intervention contributed significantly to improvements in cognitive function and functional exercise capacity in geriatric patients performing multi-component treatment intervention. I can only support this because I see these effects in my senior patients regularly. These are some of my publications on this topic. And this intervention works in such a broad way that when I found that it works against Lyme disease, I was fascinated. And after 12 years of treating borreliosis patients with chronic Lyme disease patients, I published this book, which describes my experience in treating chronic borreliosis patients, neuroborreliosis patients. This book is available on Amazon. You can easily download it, buy it for your computer or in paperback. And there are many mechanisms described in detail. Summary. We can summarize the protective effects of hypoxia, hypoxic intervention, hypoxic preconditioning. I can omit here hyperoxic intervention because, as I believe, on the molecular level, they are extremely similar. Molecularly, they are undistinguishable. For mitochondria, they are not that much distinguishable. We see here that without preconditioning, ischemia or hypoxia, acute ischemia, so let it be stroke or myocardial infarction, cause immediately damage to the cellular mitochondrial and cells and results in injury phenotype. But if preconditioning was used before, this preconditioning stimulus creates a powerful antioxidative defense and also increases cellular resistance to hypoxia. Hypoxic episodes can be tolerated much longer. For instance, instead of 7 minutes, cells adapted to hypoxia can tolerate up to half an hour. So this schema is, this chart is an explanation of both phenomena. Now technical details, a comparison between hyperbaric oxygenation technology and intermittent hypoxic-hyperoxic equipment. We can see that hyperbaric oxygenation chambers are very solid devices which are equipped with necessary controlling interfaces, and actually they are efficient, but the same effects, or even I would say in some aspect more effects, more broad scape of beneficial effects we can get from the other side, from hypoxic intervention. Here you can see our up-to-date device which produces hypoxic-hyperoxic breathing mixture controlled by computer with a biofeedback system, and this just delivered to the patient via face mask, and the whole treatment is much easier, technically easy, and of course less on the, I would say, on the magnitude less expensive than hyperbaric treatment. And we can see during the treatment, we can see physiological parameters which change under hypoxic stimulation. Here we can see the blue line is the actual oxygen content of breathing mixture. The red line, first the green line, this is actual oxygenation of hemoglobin in percentage. You see under hypoxic influences, hypoxic episode, it drops dramatically. And during reoxygenation, it normalizes. And the red line is the heart rate. It also reacts accordingly. In hyperoxia, we see tachycardia, bradycardia, and tachycardia during hypoxic pauses. And the yellow line is also very informative. It's the plethysmogram, which shows the total blood perfusion of the finger, and it reflects the physiological functional state of endothelium. What we see here is reflecting what can happen in the heart and in the brain. So this picture is extremely informative. Conclusions. Natural oscillations of PO2 at mitochondrial level can drive mitochondrial quality control mechanisms throughout embryogenesis and ontogenesis. Controlled oscillations of PO2 can improve genome stability and prolong healthy lifespan by preventing, retarding clonal expansion of selfish, mutated mitochondrial DNA. Controlled intermittent oxygenation belongs to engineered natural interventions. CIO can augment mitochondrial quality control in adults and seniors. CIO can be artificially created by intermittent hypoxic-hyperoxic training and by hyperbaric oxygenation. Regarding its preventative, curative, and rehabilitative applications, the IHHT compared to HBOT is safer, more efficient, and more convenient. Nevertheless, both interventions can synergize in creation of individualized treatment protocols in humans of all ages. Thank you for your attention and your participation.
Thank you so much for that interesting, comprehensive, and insightful keynote lecture, Dr. Prokopov, as well as for your in-depth assessment of these matters. It was truly enlightening. As this is just
Thank you for inviting me.
Our pleasure entirely. And as this is just the first installment in an ongoing planned series of webinars focusing on analyzing the results of recent practical applications in healthy human longevity and the translation from theory into practice, I'm sure we'll have an opportunity to bring you on again to discuss related topics in the near future. We're very much looking forward to that. And we'd like to thank you once again, Dr. Prokopov, for joining us today.
Thank you very much.